all, I'm Sayed Kashaf and today I'm going to conduct the session on locomotion and movement. So first of all, we will understand the difference between locomotion and movement. So we all know that movement is one of the significant feature of the living organism, and animal as well as many plants and some lower organisms, they all exhibits the wide range of movement, right? So this movement is when the living organism moves a body part or a part to bring without a change in the position is known as movement. Whereas the locomotion, when the change in the position and the location occurs by the organism, that is known as locomotion. Means when I change the posture by sitting in a confined area, that is known as movement. If I'm moving my head or a neck and changing my posture through my hands and limbs, so those are movements. But locomotion, when I change my position from one place to another, that is known as locomotion. So this locomotion, all locomotions are movement. One of the most important point in NCERT textbook, it has written that all locomotions are movement, but all movements are not locomotion. So both of these are brought about by the joint effort of skeletal muscle and muscular system. So the movement is seen in both vertebrates and invertebrates. So we will discuss about the type of movement, right? So here we are talking about the higher organism like a human. So we are mainly focusing on the human based movement only. So the cell of the human body exhibit three main type of movement, namely amoeboid, ciliary and muscular. So first of all, we will discuss about the amoeboid movement. This amoeboid movement, it is a most common mode of locomotion. It is like crawling type of the movement, which is accomplished with the protrusion of the cytoplasm of the cell, which are involved in the formation of pseudopodia, which is a false feet, right? So this amoeboid movement, now the question occurs, what type of cells exhibit this amoeboid movement in a human body? So the answer of this question is very simple, that the cell which exhibits this amoeboid movement in human body are the WBCs. So what happened when the WBC squeeze out from the blood capillaries into the tissue, that squeezing mechanism, that squeezing phenomena is known as diapedesis, and such WBCs are neutrophils, macrophages, right? So now the another one is ciliary movement. It is brought about by the appendages called cilia. This cilia is hair-like cells that move in one direction to clean the debris and dust from the body, right? These are the microscopic and thin hair-like extension found in some cells or on the some organism. And these cilia are found in the fallopian tube as well as in our nose, in throat, in lungs, and in even in our brain. And another mode of locomotion are also seen in a human, that is flagellar movement, which is responsible for the movement of mature sperm cell, right? So another one is muscular movement. So the third one is muscular movement. They are primarily responsible for maintaining and changing the posture, locomotion, as well as the movement of the internal organ, such as contraction of the heart and the movement of the food through the digestive system via peristalsis movement, that we all know. So we know that this muscle movement is more complex movement, right? So which is brought about by the musculoskeletal system. So this type of movement is seen in higher vertebrates. So to understand more about this movement, which is brought about by this musculoskeletal system, we need to understand the type of muscles and the joints. So first we will discuss about the type of muscles. So muscles, as we all know, these muscles are the specialized tissue which are originate from the mesodermal cells, and they have special properties like excitability and contractibility, extensibility, and elasticity. So this excitability is the capacity of the skeletal muscle to respond to the stimuli, whereas this contractibility is the ability of the skeletal muscle to shorten with force. And extensibility means the skeletal muscle stretches after the contraction. And the last one is elasticity, is the ability of the skeletal muscle recoil to their original resting length after the contraction, after the stretch. So these four are the characteristic feature of the muscles that we have discussed. And this muscle, the three type of muscles are found in the higher vertebrates, that are striated muscle or a skeletal muscle, unstriated or smooth muscle, and cardiac muscle. We will see one by one in detail. So the first one is striated muscle. These muscles are voluntary in nature. As this name suggests, that says that the striations are present, means striation are present. That means it shows that there is a region of dark band and a light band, and this movement is under the control of our will, and they mainly responsible for bringing about the movement of the posture and location of the organism, and they are called striated muscle due to the presence of striation, which when we see under the microscope. These cells, these striated muscles, have multinucleus and they are unbranched. This is the microscopic view of the striated muscle. Now the second one is unstriated or smooth muscle. So this smooth muscle are known as myocyte and have fusiform shape. This smooth muscle are found in the hollow organ, right? These muscles are involuntary in nature, which means their movement is not under the control of our will. When we see under the microscope, they do not have any striation on them, means there is no light or dark band appearance. These muscles are found in the digestive tract as well as in the reproductive tract and in the respiratory tract and the uterus as well as in urinary bladder and many more. So third one is now cardiac muscle. So these cardiac muscles are specialized muscles. They are found only in the heart and they are involuntary muscle which have striation on them, but the striations are not as dark as you can found in the skeletal muscle, and they have property to contract and relax in a rhythmic manner, right? And the characteristic feature of this cardiac muscle is they have they are like they are involuntary, they are uninucleated and they are branched. You can see the branching pattern appears in the cardiac muscle. And one more important characteristic of this cardiac muscle is the intercalated disc. Means this intercalated discs are made up of two type of junction, which is known as gap junction and desmosome. So this gap junction is this gap junction helps in the exchange of components from one cell to another, whereas this desmosome helps in the attachment. So now that was all about the muscles. Now we will talk about the type of muscle according to the action. So many muscles are named by the action the muscle they perform. So these include flexor, extensor, abductor, adductor, levator, depressor, supinator and pronator, spinster, tensor and rotator. We will see one by one. This flexor muscle, flexor muscle decreases the anterior angle at the joint and extensor increases the anterior angle of the joint. You can see the angle between these two arms is wider than the flexor. It is nearly up to 160 and it is nearly up to 90 degrees. Now the second one is abductor, which moves a bone away from the midline from the vertebral column, and the abductor moves a bone closer to the midline. You can see as we raise our hand up, this muscle moves away from the midline, and as we put our hand back to its original position, this muscle comes in a relaxed position or else in its original position. Now the another one is levator, which raises the structure, and depressor moves the structure down. Supinator turns the palm of the hand up, and the pronator turns the palm down. And the sphincter decreases the size of the opening. We all know that sphincter muscles are found in the stomach, then esophagus, and in digestive tract also. So this sphincter muscles helps in the contraction and relaxation process. So now the tensor tenses a body, and rotator turns a bone around it exists. Now, as we are learning the locomotion and the movement, our prime objective is to learn the skeletal muscle, right? So this skeletal muscle is a muscle tissue that is attached to the bone, and it is involved in the functioning of the different part of the body. And these muscles are also called as voluntary muscles, as they come under the control of our nervous system. So as we know that this skeletal muscle are also known as striated muscle, which contains a dark and light band, and these skeletal muscles are attached to the bones by a bundle of collagen fiber known as tendons. So now this skeletal muscle, first we will understand this image, then we will study that slide again. So first this is the transverse section of the muscle, and the outermost layer of the muscle, which is made up of dense connective tissue, which covers the entire muscle, known as epimysium. This is the muscle which covers this, and this connective tissue which covers the muscle is known as epimysium. And inside this muscle, there are small unit of muscles are present in the form of bundles. These are the small bundles known as fasciculi or a muscle bundle. And this fasciculi are again has another covering, which is also made up of dense connective tissue known as perimysium. And this and each muscle bundle are attached with each other via a collagenous bundle, which connects the muscle bundle. These collagenous bundle are known as fascia. This red color region is a collagenous tissue, which connects the one fascicle with the another, and this region is known as fascia. And this fasciculi contains small many muscle fiber, and this and the covering of this muscle fiber are known as endomysium, which is made up of areolar connective tissue, which is loose connective tissue. So this muscle fiber, so this is the enhanced structure of the muscle fiber or a muscle cell. So muscle cell is the plasma membrane of the muscle cell is known as sarcolemma, and the cytoplasm of the muscle cell is known as sarcoplasma, right? And there is a strip-like patterns which are arranged alternatively. Some of the region looks dark and some region looks light. This strip-like appearance is known as myofibrils. So when we magnify this myofibril, we will come to know that there is another structure known as sarcomere. It is a functional unit of the muscle. So this muscle fiber contains sarcoplasmic reticulum, which surrounds the myofibril and holds a reservoir of the calcium ion needed to cause muscle contraction. Then there is another tubular enfolding known as transverse tubuli. This is the tubular enfolding, which is known as T-tubuli or a transverse tubuli. This T-tubuli provides a pathway for the action potential to signal the sarcoplasmic reticulum and to release the calcium ion and causing the muscle contraction. So here you can see the connective tissue is present in all the muscle, that is fascia. This is fascia, and enclosing each muscle layer of connective tissue is known as epimysium, and enclosing each fascicle is a layer called perimysium, and enclosing each muscle fiber is a layer of connective tissue called endomysium. This we have discussed right now, right? And as we all know that the muscle fiber or the muscle cells are formed by the fusion of developmental myoblasts in the process known as myogenesis, and the muscle fiber are the cylindrical and they have more than one nuclei. As we all know that this skeletal muscle are multinucleated or syncytial cells, and they also have multiple mitochondria to meet their energy needs, right? Because they have to perform the contraction mechanism, so they require high amount of ATP. So this ATP can be served by this mitochondria. And the muscle fiber are in turn composed of myofibrils, right? And these myofibrils are composed of actin and myosin filaments. You can see the dark and the light band, and the repeated unit of sarcomere. So which are the basic functional unit of the muscle that we have discussed. So the question arises, what are the sarcomere? So before that, this is the only image that I have tried to draw in a resolved form. You can see this epimysium which covers this muscle and small bundles known as fascicle, and this fascicular again have a outer covering known as perimysium, and inside that fascicle there are small unit of muscle fiber or a cell which has again the another covering known as endomysium. These are tendons which so the muscle which attach with the bone via collagenous tissue known as tendons. So now question arises, what is sarcomere, or which region we call the sarcomere? So the portion of the myofibril between the two successive Z line is considered as a functional unit of the contraction, that is sarcomere. These are the two Z lines, and this is the entire region of the sarcomere. So as we all know that myofibrils has a striated appearance, right? Because of the presence of two important protein, that is actin and myosin, and the light band contain the actin, that is I band, right? This I band, and this is also known as isotropic band, whereas the dark band called A or anisotropic band, which contain myosin. This is the thick filament which is known as myosin. So this actin filament is thinner as compared to this myosin filament, and this in the center of this each I band, you can see the center of each I band is an elastic fiber called Z line, which bisects it, and the thin filament are firmly attached to the Z line. As we all know that actin is a thin filament, so this round structure is known as actin filament, which is firmly attached with the Z line, and the thick filament in the A band are also held together in the middle of this band by thin fibrous membrane called M line. This M line is not shown here, but you can assume that this middle region is the M line, and A and I band are arranged alternatively throughout the length of this myofibrils. So you can understand by this picture that this region is the centralized region is the M line, and at which this myosin is present in the A band. Whereas this central part of the thick filament, which is not overlapped by thin filament, that is called H zone. You can see that there is no presence of any actin filament at this central position. So this was all about the structure of this sarcomere, right? Now we will understand this. We have discussed about the muscle and the structure. Now we will discuss that how this muscle contraction takes place and how many proteins are responsible for the contraction. So structure of the contractile protein. First we will study this. So there are two types of protein. One is contractile protein and another one is regulatory protein. So this contractile protein are of two types, actin and myosin, whereas this regulatory protein are also of two types, tropomyosin and troponin. So as the name suggests that this contractile protein are participating in the contraction, whereas this regulatory protein participates in a switch on or off mechanism of the contraction, right? So this regulatory protein, the first one is tropomyosin, So this tropomyosin is present on the actin, which covers the myosin binding site. So the tropomyosin present on the actin in the relaxed state and favors the switch off mechanism of contraction. In short, this tropomyosin is a filament which masks the active site of the for binding the myosin on the actin during relaxed state. Whereas this troponin is a trimeric protein which contains troponin I, troponin T, and troponin C. So this troponin I, which inhibits the removal of tropomyosin, troponin T, which binds with the tropomyosin, and troponin C is which binds with the calcium ion, because we all know that the calcium ions are required for the contraction, right? So now we will discuss the structure. Here is the actin filament. This actin filament is made up of 2F filament, 2F actin filament, which is helically arranged, right? And each actin filament is a polymer of monomeric G or a globular actin. So this green, round, small balls are known as globular actin. When these globular actin polymerizes, they form F actin. So 2F actin, 2F filaments, they helically wound each other and forms a actin filament, upon which there is another 2 filament of another protein known as tropomyosin, also run together of the F actin throughout its length. Right, this brownish thread-like structure known as tropomyosin, and a complex protein, as we all know, troponin, is distributed at the regular interval on the tropomyosin. You can see this troponin, we all know that troponin is a trimeric protein. So this small balls is a troponin that contain 3 components, troponin I, troponin T, and troponin C, which are arranged at the regular interval of this tropomyosin. Now another one is, now this was all about the actin filament. Now we will discuss about the myosin filament. So each myosin filament is also a polymerized protein, and many monomeric protein called meromyosin and constitute one thick filament. So we all know that this meromyosin polymerizes and form myosin, and each head with a short arm and tail forms a heavy meromyosin. This region is known as heavy meromyosin, and the tail region is known as light meromyosin. Okay. So this heavy meromyosin component, that is the head and the short arm, projects outward at the regular distance. And this meromyosin has 2 important components that are globular and the angle from each other, and the surface of this polymerized myosin filament are known as cross arm. This cross arm attached with the actin filament. And the globular head contains 2 sites. One is the ATP binding site and another one is actin binding site. Here, 2 sites, ATP binding site and actin binding site. So by looking at this structure, now we are clear with that actin has the myosin binding site and myosin has actin binding site, right? So, but this is in a relaxed state. When they will bind with each other, then the contraction will take place. So we will discuss how this contraction will take place. So the mechanism of muscle contraction. Now we are discussing the mechanism, how this contraction occurs. So the sliding filament theory explains the mechanism of the muscle contraction, which states that the contraction of the muscle fiber takes place by sliding of thin filament over the thick filament, right? You can see here, when this thin filament slide over completely over the thick filament, the contraction will take place and ultimately results in the shortening of the length of sarcomere, right? Now, how this process takes place. So the junction between the motor neuron and the sarcolemma of the muscle fiber is called as neuromuscular junction or a motor end plate. that we will discuss here. This is the neuromuscular junction, or else in short we can call it as NMJ. So how this process takes place? A neural signal released by the central nervous system when reaches the junction releases a neurotransmitter, that is acetylcholine, which generates an action potential in the sarcolemma, which spreads through the muscle fiber and causes the release of calcium ion in the sarcoplasm, as we all know. So that how this process takes place, now we will discuss in detail by visualizing this image. So as we all know that for contraction, there need to be such stimulation, right? So neurons or the nerve cells, they stimulate when the polarity across the plasma membrane changes. So this polarity change ultimately results in the action potential, generation of this action potential, and this action potential travels along the neuron until it reaches the end of the neurons, right? And a gap called synapse, this is the gap between the muscle, muscle means the sarcolemma, and the neuron. So this gap called synapse or a synaptic cleft, which separates the neuron from the muscle or another neuron. And if the neuron stimulates a muscle, then then this neuron is known as motor neuron, and it is specialized synapse in a synapses occur, that is known as neuromuscular junction. So how this process takes place, that we need to understand. So as we all know this, when this action potential generates, it releases in the acetylcholine. When the action potential of the neuron reaches the muscular junction, the neuron secretes the neurotransmitter, which diffuses across the synaptic cleft. And what happened? This action potential generated, and we know that the tubular structure. So that tubular structure receives the receptor from the motor end plate and transfer that action potential to the sarcoplasma throughout this system, so throughout that cell. Now now we all know that sarcoplasm contains sarcoplasmic reticulum, which ultimately results in the release of calcium ion, right? And the myosin cross bridge forms. How? So this calcium released by the action by this sarcoplasmic reticulum binds to the troponin molecule. We all know that troponin molecule contain three components. One is I, one is T, and one is C. So this calcium binds with the troponin molecule, and this results in the exposing the active site of the actin, which ultimately results in the binding of myosin and formation of the cross bridge. Now, how this process takes place, that we will understand. Right? So till here, I think it is clear. Now we will understand the contraction, how this contraction takes place. So as we all know, when the contraction takes place. Hello?
ब्रेक करना है क्या?
नहीं सर, हो जाएगा सर, ये लास्ट स्लाइड है, 2 स्लाइड और है. So as we all know that the actin filaments are attached with the Z line, right? So when the contraction takes place, these 2 Z lines pulls inward and thereby causing the shortening of the sarcomere, that is the contraction, and where the I bands get reduced, whereas these A band retain the length. So this myosin releases the ADP and PI and goes back to its relaxed state, and a new ATP binds and cross bridge is broken and breakage is repeated, causing the further sliding. So first, from now here, we will discuss how this crossing takes place, cross bridge takes place. So as we all know that ATP binds to the myosin because myosin contain the 2 sites. One is active actin binding site and another one is ATP binding site. So what happened when ATP binds to the myosin head? It converts the ATP into ADP and 1 inorganic phosphate, which remains attached to the myosin head here. And what another see this calcium ions, This calcium ions exposes the binding site of the actin filament, right? We all know that calcium ions binds with the troponin C molecule, right? And this calcium, when this calcium ions binds with the troponin, this results in the removal of the tropomyosin and expose the position of the actin filament for the attachment of the myosin head, right? So here this myosin head is going to attach with this active site which is present on the actin filament. So when the attachment of the actin, when this attachment sites are exposed, this myosin head will bind and cross bridge will takes place, right? So what happened? Now what happened, as the cross bridge will takes place, this ADP and PI will get released and results in the sliding motion of the actin, right? It will contract, and the attachment of the cross bridge between the myosin and the actin causes the release of the ADP. You can see this ADP and PI is now released from the head of this myosin, and this in turn changes the causes the change in the shape of this myosin head, which generates the sliding movement of the actin towards the center of the sarcomere, right? And this pulls the 2Z disc together, as we have discussed. This pulls this 2Z disc together and effectively contract the muscle fiber to produce a power stroke. And now what happened? A new ATP molecule will attach to this myosin head, and a cross bridge between the actin and the myosin will get break and returning to its original position of the myosin head, which is ultimately results in the unattached head. So what happens? Sometimes what happens? Our muscles get stiff. It means that when it was contracted, now there is no availability of the free ATP molecule that will detach this contraction. So that will ultimately result in the stiffness of the muscle, right? So this was all about the contraction and the mechanism, how this mechanism and the contraction takes place. Now the another one is muscle. So this muscle, as we all know, the muscle appears red in color due to the presence of storing pigment known as myoglobin, and the red fiber contain plenty of mitochondria which can utilize a large amount of oxygen stored, and such muscles are called red fibers. And this myoglobin content is high in some of the muscle, which gives reddish appearance, right? Because this pigment is red in color, and in the form of ATP production that can also be called as aerobic muscles. Some muscle possess very less quantity of myoglobin and therefore appear pale or whitish in color. Those fibers are known as white fibers, and the number of the mitochondria are few in white fibers, but the amount of sarcoplasmic reticulum is high, and they depend on the anaerobic process of energy. Thank you.